there's not much more to say. I hope you enjoy the lightning talks, and see you later on Got the Town.
Yes, see you in a few minutes.
Hi everyone, my name is Eddie, and I am really excited about being here with all of you guys attending the 2021 Unique Student Symposium. In a moment, we will have the opportunity to briefly enter the research universe of 9 of our fellow neuro-AI student researchers. But before that, let me just take a moment to explain to you how this is going to work. So first, we will see each of the pre-recorded 3-minute videos that were selected for this event. And then, before our next keynote speaker's presentation, you will have the opportunity to go ahead and meet with these colleagues to discuss their research and ask them your question at their individual booth in the showroom of our Gathertown environment. So speakers, don't miss this opportunity to get feedback from your peers. And attendees, please don't be shy and enjoy this session to discuss and socialize. So without more delay, here are the USS 2021 lightning talks.
Hello everyone, I'm very excited to be here to talk about my work on developing holistic predictive models of cortical responses to naturalistic stimuli. This talk is going to focus on computational models that can predict how the brain will respond when presented with naturalistic audiovisual stimuli. So here's the setup. Subjects passively watch movies inside a scanner while their brain responses are being recorded with functional MRI. In this work, we model these brain responses using deep neural networks to understand how the brain processes this incoming sensory information with 2 goals in mind. First, we want to build better models that capture brain activity more stringently than existing approaches. Second, we also want to probe these models to elevate our understanding of the brain. Here is the illustration of our approach. The simplest model we use is a unimodal model that predicts neural responses using single snapshots of the sensory signals, either input images or audio signals represented in the form of a spectrogram and fed through a hierarchical convolutional neural network. We also train 20-sec versions of these models that extract the same features from a sequence of 20 input images or spectrograms, but uses a recurrent network to dynamically recompress the input sequences. Finally, we have a multimodal version of these models that use auditory and visual features jointly to make predictions. By varying the amount and type of information used by different encoding models and comparing the resulting prediction performance, we can characterize how neural resources are distributed across the cortex for processing naturalistic video sequences. In our first set of results, we computed the mean prediction accuracy in 5 groups of regions. The first 2 groups, comprising auditory and visual regions separately, do not show marked improvements from using multisensory inputs. The performance boost with multisensory and longer timescale inputs is more pronounced in the remaining 3 groups, which are not preferentially associated with a single modality and are involved in higher order processing of sensory information, namely known multisensory sites, language processing areas, and regions situated in the frontal cortex. Another interesting observation we make is regarding the timescales. Using prediction performance alone as a metric, we can discern a temporal hierarchy within the auditory cortex, as there is a small contribution of longer timescale inputs on prediction accuracy within early auditory areas, and a much larger improvement is seen for the higher auditory regions. Following a similar approach, we observe that the performance gap is higher between dorsal stream areas, whereas no difference is seen across the ventral vision areas, consistent with the known association of the dorsal stream with action-oriented behaviors and ventral stream with object recognition. Next, we want to investigate the possibility of using encoding models as neural activity synthesizers. Given that we have a predictive model linking stimulus and brain activity, we can feed it stimuli from alternative paradigms such as typical stimuli from task fMRI experiments and test how well these models transfer to this new domain. In this experiment, we predict the brain activity for different images and then use the predicted activations to generate the contrasts for 2 well-studied visual categories, namely faces and places. And we find these models, despite having only seen naturalistic image data during training, can indeed generalize remarkably to these synthetic images and replicate the contrast maps for processing faces and scenes. To conclude, using just 6 encoding models trained only on nearly half an hour of naturalistic data per subject, we can replicate findings from a large number of prior studies on sensory processing. Specifically, a single naturalistic FMRI experiment helped us characterize the functional modules involved in facial and scene processing. It helped us distinguish the temporal hierarchy and reproduce the multimodal architecture of the brain. Hopefully, this convinces you that the interplay of naturalistic neuroimaging and predictive models can be very exciting in neuroscience.
Hello, my name is WIESNER and I'm a PhD student in biophotonics under the supervision of Flavie Lava-Cardinal and Paul Lecon. In the lab, we wonder how learning and memory is directed by the molecular rearrangement of synaptic proteins at the synapse. Initially, there is calcium coming into the synapse, which serves as a second messenger to really recruit these different synaptic proteins. We therefore aim to characterize the calcium dynamics in the synapse population under different plasticity paradigms. We use dissociated hippocampal cultures and transfect them with a calcium sensor GCAM. What you see here is really these localized fluctuations in intensity. It's really calcium coming into the synapses. So we call these miniature synaptic calcium transients. To analyze these, we use the 3D unit approach to detect and segment the calcium transients. And we found that this approach really significantly detects more miniature synaptic calcium transients of smaller amplitudes. Furthermore, it also has a smoother segmentation than a generally used intensity-based algorithm. We then induce synaptic plasticity in our model culture by using a chemical RTP paradigm, and we analyze these by characterizing the different regions that show calcium transients. We first saw regions that have calcium transients only before a CRTP paradigm, so we call them silenced regions. Then we have regions that show calcium transients before and after, so they are recurrent regions, and regions that are only getting recruited by the activity paradigm, so new regions. The different regions also show different characteristics in their calcium transients. New and silent regions have generally smaller amplitude calcium transients than recurrent regions, and also the volume of these calcium transients is similar. We then also saw that the shape factor indicates that the calcium transients of recurrent regions are really spreading into the synaptic shaft, while silenced and new regions are calcium transients that stay compartmentalized within the synapse. Then we saw that when we calculated how many of these regions can be found on the dendritic shaft, that the CRTP paradigm really recruits new regions which we cannot observe in the control. We then used dimension reduction and hierarchical clustering to identify calcium transient subtypes independent on the region they occurred. And we found 4 major calcium transient subtypes, which are really recruited by the CRTP paradigm that have a larger volume and a larger amplitude. With that, I would like to conclude that we use optical recording and machine learning assisted analysis to characterize the diversity of calcium transients and to further understand how neurons integrate information. Thank you very much for your attention, and I'm looking forward to your questions.
Hello, everyone. My name is DONGYAN. I'm a PhD candidate at Mila and McGill University here in Montreal with Blake Richards. Today, I'll be presenting some data on the representations of space, time, and memory in deep reinforcement learning agents. Our experiment is inspired by the delayed non-matched sample task typically used to test working memory in rodents. The animal is placed in a triangular chamber with a touch screen. In each trial, the animal receives a sample location on the touch screen, either on the left or on the right. The animal will experience an interval during which it has to remember the location of the sample. After the interval, the animal will be given 2 choices on the touch screen, and it has to choose the non-matched location to receive a reward. Our calcium imaging data from hippocampus C81 in mice suggested that neurons fire at consecutive moments during the delayed period, commonly referred to as time cells. Here, in both panels, the cells are sorted according to when they fire during the interval after the left sample. We can see that each cell have different activity patterns for left and right samples, suggesting that these firing sequences might serve as a neural substrate for maintaining working memory. And here is after we sort the firing sequence after the right sample as well. So we simulated this task in a video game-like environment and trained an actor-critic network with a convolutional encoder and a recurrent controller on this task. We recorded the hidden states of the LSTM layer throughout training. We discovered that during the mnemonic delay, the LSTM units in the artificial neural network also exhibited the time cell phenomenon, and different samples locations elicit different activation sequences in these LSTM units. Again, the cells are sorted according to the firing order following the left sample, and here is after we sort the cells following the right sample. Moreover, some LSTM cells exhibited activity patterns similar to place cells and splitter cells observed in rodent hippocampus. Here's an example of splitter unit whose hidden state activities are correlated with agent's location. These splitter activities emerge naturally with or without memory demand and encode the sample. To summarize, our deep reinforcement learning network trained on working memory task exhibited spatial temporal representations similar to those observed in rodents. These representations may serve as a substrate to encode stimulus information during the mnemonic delay. Our results provided a computational model for the emerging spatial temporal representations in the brain on an algorithmic level. And lastly, I just want to say thank you to my lab, our collaborators, and our funding agencies for supporting our research.
My name is ZITONG Lu, a master's student in cognitive neuroscience at East China Normal University, and I will be a PhD student at Ohio State University this fall. Today, my talk is about representation comparisons between human brains and hierarchical deep convolutional neural network in face perception review of fatigue mechanism of repetition suppression. In our daily life, the brain can process various facial information in a very short time, and sometimes you may see the same face again. Some previous research found that the neural activities would decrease when you see the same face repeatedly. We call it repetition suppression. However, the primary neural mechanism of repetition suppression in our brains is still unclear. For example, fatigue or shuffling of neuronal activities. As we know, the deep convolutional neural network, DCNN, can achieve the performance of face recognition at a human level. So here we use DCNN as a tool to explore the neural mechanism of repetition suppression and compare the representations between the human brain and the DCNN. Here we use the EEG data from an open dataset. In this experiment, there are 3 types of faces and 3 test conditions. Participants would see each face twice. New means the first time you see this face. Immediate means you see the face again immediately. Delay means you see it again after several trials. For the brain representation, we use EEG classification-based decoding method to construct time-by-time RDMs. The shape of each RDM is 9 by 9. And for the DCNN representations, we input all images to the DCNN, and we can get DCNN RDMs for each layer. And we constructed 2 representation suppression models, fatigue model and shuffling model, to modify the activation of DCNN. In fatigue model, the activation of neurons with stronger response to face stimulus were attenuated. In shuffling model, the less responsive neuron didn't respond. So based on the modification, we can get bigger size RDMs for DCN. Then we conducted the cross-model representation similarity analysis. The results show that in layer 16, the representations of human brain were more similar to those in fatigue-modified DCN. Also, human brain shows stronger and longer similarities with the higher layer in fatigue-modified DCN. In summary, the current research supports the fatigue mechanism as a more flexible neural mechanism of facial repetition suppression, and the comparison between representations in human brain and hierarchical DCN provides a promising tool to simulate and infer the brain mechanism underlying human behaviors. Thank you.
Hello, my name is Miriam Han, and I'm an undergraduate researcher at BioPT Lab at McGill under the supervision of Dr. Stephanie Blaine Morris and PhD candidate Charlotte Mashk. Thank you for this opportunity to share my passion for this research with fellow scientists. After patients experience a traumatic brain injury, they're often diagnosed with disorder of consciousness. These disorders are characterized by impaired wakefulness and awareness, and specifically can be divided into 3 categories, depending on their level of self-awareness and environmental awareness. It is difficult to accurately assess levels of consciousness and predict the prognosis of patients with DOC because their behavioral responses are often inconsistent and ambiguous. Currently, there are inevitably high rates of inaccuracy in the diagnosis and prediction of recovery, which hinder physicians from providing the best treatment, setting realistic goals of recovery, and making ethical decisions regarding the patient. This leads to our first main takeaway, which is the importance in establishing a non-task dependent measure that does not require behavioral responsiveness to assess covert consciousness in DOC patients and their potential to recover consciousness. I would like to now move on to the second takeaway, which is that reconfiguration between anesthetized and baseline state can be used to distinguish the patient's potential for recovery of consciousness using general anesthetic such as isoflurane and propofol. In this case study, 5 minutes of resting state EEG data was recorded from 1 healthy participant and 2 brain injured patients during the baseline and anesthetized states. The EEG data were analyzed to characterize each state in alpha and theta frequencies. The first feature of interest was the topographic location of the hub during the sedation and baseline states of subjects. And the second was the directionality of functional connectivity measured in DPLI. So the healthy participants showed that during the anesthesia, there was an interiorization of alpha hub, and in the directionality of the functional connectivity, there was decrease and reconfiguration in the change of the direction. In comparison to the healthy brain, the recovered brain patient had the similar changes observed during baseline, while the unrecovered showed static brain. If we move on to directionality of functional connectivity, again, the recovered patient showed a reconfiguration between these 2 states, while the unrecovered patient did not. This study represents a method of assessment that is robust, generalizable, and easily translatable in routine clinical practice. It holds great potential in developing techniques to uncover reconfiguration patterns in brain injured and DOC patients to improve accuracy and confidence in physicians' assessment.
Stroke is a brain disease that causes neuronal and functional loss. Largely over the time, some sort of recovery or compensation from the loss of function can be seen. Contemporary approaches are limited to exercise and training. To study this in stroke models of mice, here we are proposing a brain-computer interface to map their brain activity with the sensory feedback. So this is my current work at Dr. Murphy's lab. My name is PANKAJ, and I'm going to talk about real-time sensory feedback of cortical activity with a BCI. We are employing transgenic mice expressing cortex-wide GCAM6. Here is a view of mouse whole brain in 3D by Murakami et al. 2018. And we are imaging the dorsal projection of such brain. Our BCI setup involves head-fixed mice. Raw brain images, as you see in green, are processed in real time. We can choose cortical regions R1 and R2 as you see overlaid on the activation map. The relative activity of R1 minus R2 is mapped to an auditory sensory feedback where tone frequency depends on the relative ROI activations. We can impose any other arbitrary rules for mapping activations in these ROI to the auditory tone feedback as well. We also set a pre-computed threshold above which the mice are rewarded with a drop of water. The overall sensory feedback delay is typically around 100 milliseconds in this system. With this setup, we did the following experiment. We implant windows on day 0, let them recover for a week, Then we start habituation and move on to closed-loop feedback training for 15 days. With this system, we train 19 mice involving different target ROIs. We find that normalized number of rewards significantly increasing over the days. We trained several mice with different target regions and different rules. In this figure, each row represents the reward-centered brain activations for different target regions and the associated rule during the training, as labeled on the left of the image. In conclusion, we present a real-time, low-cost, GCAM-based optical BCI where mice can be trained to modulate their brain activity in target brain regions. Using this, we can assess and probe different brain regions quickly and determine optimal parameters for BCI control. In future, we are trying to assess efficacy of this system for stroke recovery training. We are also integrating this BCI into the in-house home cages for long-term study. Additionally, we would like to test this system on different indicators as well as on multicolor expressions in the brain. With that, I thank you for your time and attention.
The brain is a highly complex organ making complex decisions and computations every millisecond. This is made possible by billions of neurons, each fundamentally different in the types of intrinsic dynamics they exhibit. Some exhibit Lie integration of currents with threshold-based spiking, and others exhibit even more complex types of dynamics such as threshold adaptation and after-spike currents. When we compare this to current artificial neural networks, or ANNs, that attempt to emulate the brain's computing powers, we find that although ANNs are very successful at tasks like image recognition and sentence translation, they lack biological realism and they lack diversity in neuronal dynamics. This raises the question of what advantage do the diversity and complexity of dynamics we see in the brain have? The goal of our research is to assess the role of complex and diverse dynamics in network computation by implementing them in ANNs. We start by developing a neuronal model that implements biologically realistic dynamics, namely leaky integrated fire dynamics and after-spike currents. This model can also learn parameters underlying these dynamics using gradient descent. We optimize a network of these neurons on a queued pattern generation task. In this task, networks receive inputs from a series of input sources, out of which only a subset is active during a trial. The network is trained to produce a sinusoid whose frequency is based on the input sources. We set up a network with a single recurrent hidden layer, which is initialized with no diversity. Over training, the network can learn both neuronal parameters in these neurons and synaptic weights. Indeed, this network was able to learn this task, and as shown here, it produced different frequency sinusoids based on the input. Interestingly, after-spike currents were required to learn this task successfully. We then characterized the diversity learned by the network. We first analyzed the distribution of each learnable parameter across neurons in the trained network, and we observed a diversity in both membrane-related parameters and in after-spike current parameters. Next, we presented each neuron in the trained network with constant input current and observed diversity in response types across the network. These two findings support that the diversity in dynamics was learned in this training and was beneficial to the network. In the future, we want to assess the biological realism of the learned diversity by comparing parameters learned by our model to those found in the brain. Thank you all for listening.
Hi, my name is Jonathan CORNFORD. I'm going to present our work building artificial neural networks for separate populations of excitatory and inhibitory units, which is a constraint commonly referred to as Dale's principle. Also, this work was just accepted at ICLR, so please visit Open Review for the full paper if you're interested. Okay, so as we know, ANNs are increasingly being used in neuroscience. And from a neuroscientific perspective, the ideal network is one that is as biologically plausible as possible without sacrificing learning performance. And so there are a number of reasons for this. For example, for the task of modeling brain representations, is we found that the similarity of ANN and brain representations correlates with both task performance and the biological realism of the architecture. And I think from a more practical perspective, biologically motivated architectures can just help facilitate cross-disciplinary research. between neuroscience and AI. Although there are a number of differences between ANNs and biological networks, one of the main differences that hasn't received that much attention is the absence of Dale's principle in artificial neural networks. So Dale's principle means that each unit can only have positive or negative output weights, and also that these weights cannot change after initialization. And obviously, a normal MLP or normal artificial neural networks, this isn't true, and units can have both positive and negative output weights. Okay, so currently the standard way to include Dale's principle in ANNs is just to constrain the sign of entire columns of the weight matrix. However, unfortunately, networks with column-constrained weight matrices don't learn very well. So here we're training an MLP on MNIST with stochastic gradient descent, and we find that they don't converge to zero training error, and they also generalize poorly. And so in our work, we propose an alternative architecture that includes Dale's principle, which takes inspiration from feed-forward inhibition in the brain. And here we place inhibition between layers of the network. And we also, like inhibition in the brain, we model inhibition as being both subtractive and divisive. Next, we use two key insights to get DANs to work as well as standard ANNs. First, we initialize the inhibition such that it normalizes the excitatory input to the next layer. And second, this architecture introduces disparities in the amount that updates to different parameter types affects the network. And so we therefore scale down updates to inhibitory parameters to account for this. Okay, so returning to the same figure from before when we were testing column EIs on MNIST, we can see that the DAN architecture in orange performs much better than the column EI in green, and as well as the standard MLP in black. It also performs pretty well with just one inhibitory unit per layer, shown in blue, and we can also see the importance of the update corrections in gray. So in the paper, we also test DANs on K and fashion MNIST, and we provide preliminary results extending DANs to convolutional networks on CIFAR10. So unfortunately, that's all I have time for, but if you have any questions, please feel free to email me, and thank you for listening.
Hello. I am researching task general approaches to neural dynamical modeling. Neural dynamical models, or NDM, represent neural activity as a dynamical system. NDM approaches are commonly used to relate neural activity and subject motor behavior, and to generate accurate neural activity estimates for signal reconstruction and prediction. Shown here is an example NDM paired to a closed-loop BCI, where neural activity estimates and subject behavioral information produced from the NDM is used to augment the BCI decoder. Prior NDM research has been limited to modeling neural activity collected during a single subject behavior or small set of subject tasks. This has so far limited NDM models from behavioral generalization, or the ability to model the dynamics of neural activity collected over a wide range of subject behaviors. I am creating a task general NDM by training recurrent autoencoder models to reconstruct ECOG data collected during unconstrained subject behavior. Specifically, I've trained a modified version of LFADS, a particular RNN-based generative autoencoder, to reconstruct motor and premotor cortical macaque micro-ECOG signals over a multi-day free behavior period. Example reconstructions of 200 millisecond ECOG traces are shown on the right in orange, overlaid on their target sequences, which are shown in blue. An AR model trained to reconstruct the same training set is shown in green. One can see that the autoencoder outperforms the linear method and generates accurate reconstructions of the input sequences. However, the model's performance appears to be frequency dependent, reconstructing slower components of the ECOG data more accurately than fast components. To better characterize the relationship between reconstruction bandwidth and accuracy with this model, I compared the power spectral densities of model outputs to the PSDs of target sequences. I found that model performance was in fact band limited to lower frequencies. Wider bandwidth outputs led to more accurate reconstructions, as shown on the left. Our explorations over model architectures have shown us that output bandwidth and reconstruction accuracy are primarily dependent on the width of the model RNNs, i.e. the number of hidden units in each RNN cell. Given the apparent dependence of reconstruction performance on RNN width, my next steps are to produce a new autoencoder architectures that more readily produce broadband reconstructions without as strict of a dependence on model parameter scale. This is important for creating NDMs that produce maximally accurate reconstructions of neural activity. Two considered approaches are one, the use of multiple parallel modified LFAD cells on band-limited filtrations of target ECOG sequences, and 2, augmenting the model with input and output convolutional cells. This work has been carried out with the support of my advisors, Amy Yorsborn and Ellie Schleserman. I also acknowledge these funding sources and fellowships. Please reach out to me about my work at the email address shown below. Thank you.
All right, thank you everyone who submitted a video, a talk. It was very interesting to hear all of your work. So right now we're going to move on to Gather Town for a quick social break. So if you want to go ask question to the speakers, you can go see them at their booth. And as well, there's going to be a quick active break on the beach in the town around 3:05 if you want to join and move a little for 5 minutes for the next keynote talk. See you on Gather Town.